Hello students who want to succeed. Welcome to SEPAM, Super Easy Physics and Mathematics. This video is going to help you to perform better in your biology. This part 1 of the video is going to contain question 1 to 20 of the IGCSE Biology Paper 2 MCQ extended, and we are looking at the February-March 2023 edition. The total duration for this paper is 45 minutes. I want you to take your time to go through this particular video because I'm going to be giving you tips and then explaining some of the concepts that were examined, tested in this particular paper in detail. So it's going to be a revision edition. So just take your time to go through it. So question 1 of this paper: what is used as a means of classification? What is used as a means of classification? In biology, there are 3 basic means of classification. The first one that we are used to is the use of morphology, the external feature of the organism. Then the second means of classification is the use of anatomy, which is the internal structure of the organism. But the third means of classification is what is being tested in this question. So let me just explain it so that you can understand it better. Now, the first thing we need to understand this question, some basic technical term, is the DNA. DNA is the short form for deoxyribonucleic acid. So I'm going to explain what it's all about. Now, in this deoxyribonucleic acid, it contains a sugar with 5 carbon atoms that is called deoxyribose. Now, biology compare the sequence of base from 2 different species of DNA. And the more similar the base sequence, the more closely related the species are. So this is the basic fact here. But what are the base? So one thing I want you to know is that a molecule of DNA is made up of 2 strands of nucleotides, making it a polynucleotide. And each nucleotide contains a nitrogenous base. So those nitrogenous bases contain the following: contain adenine, thymine, guanine, and cytosine. And in the base sequence, they have a pairing formula. So adenine will always pair up with thymine, and then guanine will always pair up with cytosine. So to answer this question, so that the video will not be too long, so let's go. So the answer to this question is C. Let's go to the next question. How many jointed legs does an insect have? This is a direct question. An insect has 6 legs, which are in 3 pairs. So we have 3 pairs of legs. This is a direct question. Now, the photo micrograph show a cell from a type of aquatic plant. So let's label the part shown. So you have the cytoplasm R, then you have the chloroplast. So you have the chloroplast, the green pigment that attracts light to enable plant to photosynthesize, and the chloroplast contain the chlorophyll. Then we have the cell wall. The cell wall is turgid and help to keep the plant in shape because it contains the palisade. Then we have the nucleus. which is the part that controls the activity of the plant cell. So if you look at this particular feature, which label part indicates that this plant is a plant cell? So that will be the part that is present in plant cell but not present in animal cell, which is the chlorophyll and the cell wall. So that makes the answer. So to indicate that it's a plant cell, so you have the chlorophyll and the cell wall. So the answer is D. The diagram shows a red blood cell, RBC. The actual diameter is 7 micrometer. But in this diagram, is drawn with a length of 49 millimeter. So this drawing is an enlargement of the actual. So what is the magnification of this cell based on this diagram? So the formula for magnification is the diameter of the drawing compared to the diameter of the real RBC. So we'll have that. So substituting, so you have 49 millimeter divided by 7 micrometer. So put the very unit there, then you divide. That will give you 7,000. So the answer is C. Question 5. Which molecule crosses a partially permeable membrane during osmosis? Now, let's look at the demonstration of osmosis. I just want you to understand. So if you have this an osmotic system, let's have that we have two regions. Region A, which have high concentration of water molecule. Then region B, you have low concentration of water molecule. Then you have this semi-permeable membrane. When by partially permeable means that it will allow some particles to pass through, but will not allow that. So if you look at region A, we have the big red particle, and then we have this small water molecule blue. So if they remain in this system, so we are looking at partially permeable. That means it allows some material to pass through. So if you look at this, so the water molecule pass through freely, but the red molecule cannot pass through. So this makes it semi-permeable. So if you look at it, the major factor that allows them to pass through is the size of the molecule. So if you look at all this one, for a material to be partially permeable, so the only source here based on this material that have those tiny characteristics is water. The graph shows the rate of uptake of magnesium ion by two similar plants, S and Y. Two similar plants. The root of each plant were placed in a range of solution. Each solution contained a different concentration of magnesium ion. All other conditions were kept constant. So here we have the rate of magnesium intake. Then here we have concentration of magnesium in external solution. So you see that which of the plants take more of magnesium? Plant S take more, while plant Y take less of the magnesium ion if you compare it. Now, what is the possible explanation for the difference in the result for the two plants? The answer is A. Plant Y have fewer protein carrier for magnesium ion in its cell membrane compared to plant X. So this one have more protein carrier, while this one have fewer protein carrier, because the protein carriers that help to transfer the magnesium ions. Which substance is used to test for food for vitamin C? So here we have different reagents. So I'll just talk about the reagents. So we have the dichlorophenyl iodophenol, which is DCPI. So it turns from blue color to colorless in the presence of vitamin C. So this is the reagent used for testing for vitamin C. So B is the answer. But let me just explain the use of Benedict solution and iodine solution in food tests. No, when you want to test for carbohydrates, especially the reducing carbohydrate, that is, talk about glucose or sucrose or maltose. So if you look at them, so if you have in ordinary starch, if you add Benedict solution, the color is blue. But if the starch is broken down to maltose, which is a reducing sugar, then the color will turn red. So the presence of reducing sugar, glucose, maltose, will turn the color from blue to red. Then this case here, so when you want to test for starch, adding yellow iodine, that is a natural color is of the reagent, it is yellowish. Iodine is yellowish. If you add it to a food substance that contains starch, the color turn to deep blue, so the presence of starch. So like why we use iodine to test for starch in leaf to show the presence of photosynthesis, that the leaf are photosynthesized. So I think there's also a question later that we'll talk about that tested that. So when I get that, we'll discuss it. So question 8. In DNA, what is the correct base pairing? I explained it in question 1, but let me just give you the structure of the DNA. So if you have this, a DNA has the phosphate group and then the deoxyribose group, that is a sugar, with 5 bonds. So if you look at it, so then we have the nucleotide. So we have thiamine and guanine. So this is one structure. So then in this one we pair up to form the DNA complete molecule. So if you have this, so thiamine will pair up with adenine and guanine will pair up with cytosine. So this is the answer is D. What remains at the end of an enzyme control reaction? Enzyme control reaction is a reaction that is speed up by enzyme. And then, for example, let's say sucrose. Sucrose, the enzyme that catalyzes the breakdown of sucrose is sucrase. Normally, enzymes are named based on the name of the substance that they act upon. And then it will break down sucrose to glucose and fructose. Now, but after the end of the reaction, the sucrose will be used up, and then we are left with the product, which is the glucose and the fructose, and then the enzyme will remain because it speeds up the reaction, but it's not used up in the reaction. So this is a catabolic reaction. What do you mean by catabolic? A reaction that involves breakdown of large molecule into smaller molecule. But if I combine smaller molecule to form a larger molecule, that's an anabolic reaction. So you have catabolic and anabolic. So for example, if you look at catabolic reaction, that is respiration, is a catabolic reaction. It involves the breakdown of large molecule to a small molecule. All right. So based on this, in this reaction, the sucrose is used up, so it's out of it. So we are left with the enzyme and the product. So that makes the answer A. Which feature explains the specificity of enzyme? Now, enzymes have shape that exactly fit the substance that it combined with. So when we say that enzymes are substance specific, so the answer is A, the complementary shape of active site and what substance. So one enzyme cannot act on just substance that doesn't fit into their shape. Yeah, this one I was telling you that we'll be talking about question 11, test for starch. A plant is placed in the dark until its stored starch is used up. Take note of this detail. Its stored starch is used up. The plant is placed in light with black paper over parts of one green leaf. So you have this graph paper. Now, so this part, the leaf, the part that is exposed to light. So what we are testing here, we are looking at the importance of light. Light as an important essential part of photosynthesis, condition for photosynthesis. So if this paper is covered with black paper, if this leaf is covered with black paper, that means that the part that is covered, the starch that is stored in it, after about period of 8 hours, the leaf would have used, the cells would have used up the starch. So there will be no starch in those locations. Why this part that is exposed, like this window here, showing the vein and some portion of it, so after 8 hours, the leaf is tested for starch. So what is going to happen? This part that is exposed to light will test positive. This part will test positive. Then these two small window will also test for positive. But this part that is covered with paper, because there will be no starch formed in it, because the leaf were not exposed to light, then we test negative. So which feature? So look at the key. Starch is present. If starch is present, that will show you the white part. Starch is present. Then at the end, this part here that will remain white, that is not affected by the iodine. So if you test it by iodine, so we look at it, the part that will turn blue. So if you look at it, so it's the one that will feed this feature. So where starch is present is this white light, which is here. Then these two window here and this window. So the answer, so only the area that had received light go blue with iodine. So the answer is B. Question 11. The diagram show part of a cross section of a leaf. Yeah, most students fail this question because they mistake this. This, we have the epidermis. We have this black spot, which are the chloroplasts. We have the vacuole, that is this white part. Then you have the cell wall. Then, but this tissue here, this 1, 2, 3, 4, 5, 6, these are the palisade. So this arrow, this was what most of the reasons students couldn't identify it. So they named it, this is the epidermis, but the arrow is not getting to the epidermis, just on top of it, which is the cortical. So R is the cortical, S is the palisade mesophyll, and then this is the spongy mesophyll. So most students thought that this was cortical, so they took this section C, which is what? Epidermis, palisade, and spongy. But it's wrong because this is not the epidermis. Arrow is not the epidermis, it's the cortical. So the answer is A. Now, okay, feeding. The diagram shows different types of teeth. How do you know them? You look at the shape. The one that have edges on top, the flat edges, are the one for grinding. So let's identify the teeth. We have premolar, canine, molar, and incisors. So which of them are used for grinding? So you have the molar and premolar. So 1 and 3. So that is the answer. Yeah, this diagram shows assimilation of food in the small intestine. Now, the diagram shows a villus. Structure P and Q absorb different products of digestion. Which row identify the product absorbed? So let's identify the part Q, P and Q. So P is a latia, and what does the latia do? Absorb fatty acid, and then it go and empty it into the lymphatic system, so which contain the lipids, and then eventually empty into the bloodstream. Then P is the blood capillary. Then this part here, which, okay, blood capillary help to assimilate glucose. Then you have the epithelium, which absorb water and also absorb glucose. So you have the epithelial cell. So we have this here. So if you can identify the part, so the correct answer is C. Now, let me explain this. When the glucose is absorbed by the villus is carried to the liver, the glucose will be carried to the liver, and then the excess glucose is converted to glycogen and then is stored. But when the glucose is needed again, the glycogen is converted back to glucose for the body to use it. Then the enzyme that supervises that conversion is the glucagon. So let me just read about this. Now, root L. The arrow shows the movement of substance from the soil into a root L cell. So we have two arrows and it's deliberate. You have the soil particle, then you have the porous layer, then you have the structure, we have the external structure, that is the cortex. Alright, which substance move from the soil into root L cell? So we look at this arrow, deliberate, two substances. And what are those two substances? So we have water molecule and ion. So water molecule and ion are the two things that are moving. Then let me just explain something here. Xylem will transfer the water and the ion up to the leaf, while phloem carry the manufactured food in the leaf, which is the source of the manufactured glucose and the nutrient, back to the root, which will serve as the sink. So just take note, this one is a bonus tip for you. All right, let's go to the next. By which process does water escape from stomata in the leaf? Now we have active transport. What is active transport? Active transport is the movement of molecule from region of low concentration to high concentration against the concentration gradient. So this one needs energy. Energy is involved for this to take place. Then diffusion is the movement of molecule from high concentration to low concentration without a semi-permeable membrane. Then evaporation is the conversion of water molecule to gas. Then osmosis is the movement of water molecule from high concentration to low concentration through a semi-permeable membrane. Now in the stomata, there is no semi-permeable membranes, but what happens, you have an opening, so the answer is diffusion. But most students mistake it and pick evaporation, and they were wrong. Which role about the circulatory system is correct? So you have part that pump blood and part with valves. So let's just look at it. Veins have semicircular valve to maintain the directional flow of low pressure blood back to the heart. Heart also have valves, and then on the left hand side of the heart, between the left atrium and ventricle, you have the bicuspid valve. And then on the right, between the right atrium and the right ventricle, you have the tricuspid valve. So the answer is that pumps blood is the heart, and the part with valves is heart and vein. So the answer is A. Question 18. The graph shows the pressure changes that take place in the right atrium, which is the high pressure. Okay, talk about the right atrium. Then the right ventricle and the left ventricle of a human heart when the muscle wall contracts and relax. Now, what is the pressure in the right ventricle when the left ventricle is at its maximum pressure? Now, one thing you should know, the question here is testing, because they didn't tell you which part have the low pressure and which part has the high pressure. So that is actually the major thing that is being tested in this question. Now, you say, what is the pressure in the right ventricle? The right ventricle is a part that carry low pressure. Why? And the left ventricle, you say when the left ventricle is at its maximum pressure. So left ventricle carry high pressure. So this is the pressure of the left ventricle. So then the pressure here is the pressure for the right ventricle. So let's identify the peak for the left ventricle. So the peak is here, which is about 15 kilopascal. Then, so the pressure here is going to be 3 kilopascal. So the answer is C. Question 19. The sequence of amino acid in antibody enable them to complete which function? So bind to a specific antigen. So this is a direct question. We'll look at control of diseases. Question 20, which is the last question on this part 1. The table shows some of the changes that occur during breathing. Yeah, we are looking at inspiration, that is inhalation. Okay. Which changes occur to cause inspiration? So this is, we are looking at breathing in. So for you to be able to breathe in, so let's look at what happened. We are looking at the 3 major components of the respiratory system. We have the diaphragm, the external intercostal muscle, and the internal intercostal muscle. So we're going to see, will they relax or contract? That is what the question is all about. So let's look at diaphragm. For inspiration to take place, diaphragm will contract or flatten. So that helps to increase the capacity in your thoracic cavity. So increase the capacity, increase the volume. So when the diaphragm pull down, it suck air from the trachea, from the nursery, so it goes down. So then, so the diaphragm will flatten. And for the diaphragm to be able to flatten, what is going to happen? So let's look at the intercostal muscle. The external intercostal muscle is going to contract to pull up, so your rib will rise. So your chest rises, and then by so doing, so Y will also contract. The intercostal muscle contract. Then the internal intercostal muscle is going to relax, so that the space inside your thoracic cavity can increase. So we have contract, contract, relax. So you have SYR. So the answer is C. So I want you to go to part 2 to continue with from question 21 to 40. I know you are a brilliant student, that's why you are here. Just keep in touch, because more tutorial solving past questions will be uploaded in this channel so that you can keep up to date. I will appeal that you subscribe, so just subscribe, and then kindly like and comment on this video. So I want us to improve together. So what you feel we can work upon, just give your positive comment. If you like the video, comment it. If you gain new facts, comment about it. Thank you for previewing this past question with me.